Is Dr. Rohit Pappu from Washington University, St. Louis. I also would like to welcome Dr. Daniel Raleigh from Stony Brook to the panel. Dr. Raleigh will introduce the speaker and moderate Q&A session for today. Thanks, Dan. Dan, get started.
Pleasure, Rams, and thank you, Rams, and the rest of your team for continuing to organize these seminars. It's a real pleasure for me to introduce a longtime colleague and good friend of mine, Professor Pappu from Washington University in St. Louis. Rohit had completed his undergraduate studies at University of Bangalore in India, where he studied physics, mathematics, and electronics. He then moved to Boston, or more accurately, Medford, Massachusetts, where he completed a Master's of Science in Solid State Physics, and then a PhD in computational studies of protein folding at the same institution with the late Professor Weaver, who was a real pioneer in early computational models of protein folding. After completing his PhD, Rohit moved to Washington University of St. Louis, where he was a postdoctoral fellow with Jay Ponder, followed by a second postdoc at Johns Hopkins with George Rose before he returned to Washington University in St. Louis, I believe in 2001, as an assistant professor, promoted to associate professor in 2007, full professor in 2010, and he now is a distinguished professor, the Jean Barry Distinguished Professor of Engineering. He's also the director of the Center for Science and Engineering of Living Systems. He's very well known for his work in phase separation, protein folding, neurodegeneration, with particular emphasis on Huntington's disease. And I think what distinguishes Rohit is he brings a very rigorous background in polymer physics and applies that together with high-level simulations to these problems. He has a very long and successful track record of interacting quite closely with experimentalists, and his group actually has a significant experimental component on its own. So really a very impressive approach. Without further ado, I'll turn it over to Rohit, who's going to tell us about some of his work on the linkage of folding and binding on phase separations.
Thank you, Dan. And thank you, Rams, for the invitation to be part of this group and start the new year off. And it's good fun to see a lot of old friends here. I was just scrolling through the list of participants, lots of familiar names, but also a lot of new people. So hopefully we get to talk about some things that might interest you. While I may not touch upon any specific proteinopathy today, what I'm going to do is sort of introduce you to some concepts that are at the crossroads, if you will, or actually at the intersection is the better way of putting it, of the way we think about protein aggregation and misfolding, et cetera, from a very conventional standpoint, and some new ideas that have emerged from the field of protein phase separation. So the topic will touch upon sort of linkage of folding and binding, and we'll dive right in. This audience does not need an introduction to the way to think about protein stability, but it's a good place to start. So here is a conventional way of thinking about the protein folding reaction as a two-state equilibrium between a population of unfolded molecules and a population of, I see somebody has raised their hand. I'm not sure if I should be dealing with that, but
just ignore it and go ahead. Maybe.
Okay, all right, perfect. And a population of folded molecules. And we typically, in these two-state reactions, think about a free energy of unfolding, depending on your perspective, or a free energy of folding, again, depending on your frame of reference, defined as a difference between a pair of standard state free energies. What this does is, of course, always engenders the finite likelihood that, under some set of conditions, there will be a finite concentration of unfolded molecules. And should that be the case, then we've learned over the past 3+ decades, thanks to amazing work done by Chris Dobson, Sheena Radford, and several others in this audience, summarized here by Jan Zuckerman, that one can have at the unimolecular level a two-state equilibrium between folded and unfolded populations. In a cell, of course, these molecules come about due to active protein synthesis. And the idea depicted here by Zuckerman is that these unfolded molecules can get themselves into trouble or not, depending on what the purpose of being unfolded might be about. And the types of species that one thinks about are these sort of deposits that are viewed in some cases as deposits of unfolded proteins. So they go by the names of aggregates, but the key issue here to keep in mind is that the population of unfolded proteins is largely governed by the either the rate constants, but more importantly, just the folding equilibrium. And this sort of leads us to an interesting pedagogical question, really, and I'm not telling you anything that most of you haven't thought about quite actively and even more profoundly than I have. But let's just sort of set the stage by asking something of a rhetorical question, which is what can happen if the concentrations of unfolded molecules increase above some threshold concentration? So this is where we'll take a bit of a turn away from using terms like protein aggregation, because at the end of the day, these are all really phase changes where concentration is what is called the conserved order parameter. So effectively, the idea is that if the concentration of said unfolded molecules increases, there is a possible density transition that whereby the protein plus solvent mixture can separate into two coexisting phases, whereby the dense phase is some form of viscous liquid, which will go by the name of liquid-liquid phase separation. This is quite trivial, really only applies to homopolymers. One can also think about transitions that are much more realistic, whereby proteins, which are heteropolymeric molecules, and particularly unfolded molecules are still heteropolymers, cross some threshold concentration, giving rise to coexisting phases comprising not predominantly just dispersed monomers, but actually clusters of molecules that coexist with the dense phase, that really is some form of a viscoelastic fluid or material, really, that can sort of ripen to form gels and things of that nature. And that we will refer to as a density plus a networking transition. And of course, as many people well know, when there are structural transitions that accompany the density transition, then one can start to see what we think of as a symmetry breaking operation, whereby now the molecules can sort of align along particular preferred axes, giving rise to structures known as amyloids. So let's sort of take this pedantic view and sort of distill it down to sort of a particular example, superoxide dismutase. Here, what we'll do is we'll work with a molecule that's essentially monomerized by the design of certain loops. This was done first in Mikael Oliverberg's lab. And the idea is that we'll use superoxide dismutase as a model protein that when destabilized will undergo some interesting transitions. And so here are two mutations, basically. And the idea is that at low concentrations, essentially within cells, one sees dispersed versions of SOD1. But for this mutant protein, when there is some threshold concentration that is crossed, one starts to see the formation of certain types of puncta. Here is a scale bar just for calibration, and these are in HeLa cells. And what's actually quite interesting is that one can go back and query the impact of different mutations that destabilize SOD1. So here's the wild type across a range of concentration expressions. Essentially, one gets only a one-phase behavior. But then as one starts to titrate the effects of stability by different point mutations, there are different mutant-specific threshold concentrations at which one sees the appearance of different puncta. So the message then suggests that there's a connection between protein stability and intermolecular associations that give rise to these inclusions or puncta. And the question is, what is the underlying phenomenon really about? So here's the proposal that we're going to work on through a sizable fraction of today's talk. And it goes something like this. We've got well-folded molecules that are going to be defined by a hydrophobic core. We'll say that we'll call these stickers that basically are sequestered within the core of the folded state. There is an equilibrium with the unfolded state, which now leads to these cohesive interaction driving stickers being exposed to the solvent. Perhaps they don't like that very much. And depending on the concentration of these unfolded molecules, one essentially sets up the possibility of a phase equilibrium whereby the interactions between these sticker residues enable networks to form and a whole manner of interesting phase changes to be realizable. So you're probably thinking, wait a minute, this guy is just using a whole different term for what we've always recognized as hydrophobic residues, et cetera, et cetera. So this is where the integration with a lot of what has been happening in the phase separation field will hopefully arise. So let me sort of detour and tell you about what these stickers really are and why we use this language. So as many of you perhaps appreciate, the concept of phase transitions has become quite prevalent in the intracellular phase transitions literature, giving rise to things that are known as membraneless condensates. Although the underlying physics that most people use is the language of homopolymers, we and others have been trying to sort of highlight the fact that there's actually a very important sequence grammar that underlies these phase transitions that give rise to membraneless condensates. And that's the language of stickers and spacers. And what I mean by that are the fact that heteropolymeric protein-like sequences, be they intrinsically disordered proteins or unfolded states of well-folded molecules, will basically feature these blue patches, let's say, we'll call them stickers, that can engage in reversible cross-links. These can be of a variety of different types of physical associations. They could be pi-pi interactions, cation-pi interactions, complementary electrostatic interactions, or even hydrophobic associations. But the key point is that these are chemistry specific and they are reversible. And interspersed between these stickers are going to be spacer residues that are effectively impacting the solubility profiles of these molecules. So you take these two together, sort of essentially solubility determining interactions and chemistry specific interactions, you can actually encode some very interesting phase transitions into these types of molecules. So what I'll do is summarize quickly for you some lessons we've learned over the past few years by studying prion-like domains from RNA binding proteins in particular. The work I'll summarize basically is a continuing collaboration with the lab of Tanya Mittag. At various stages has involved different people. The first part of this work involves two former postdocs in Tanya's lab and a former postdoc in my lab, Alex Holthaus. So many RNA binding proteins are involved in a whole range of proteinopathies. Here is an example, HNRNPA1. The architecture typically is quite similar, featuring one or more RNA recognition motifs or modules, and generally domains that go by the name of either prion-like domains or low complexity domains or intrinsically disordered regions. And because of this sort of overload of terms, people tend to conflate all of these as being exactly the same. but they're not. So intrinsically disordered proteins or regions basically are a superset that encompass things like low complexity domains or prion-like low complexity domains. So here is the sequence of this said low complexity domain, and you can appreciate why people may have converged on such a name, because it doesn't look like a whole lot of different amino acids are being utilized. There appear to be a surfeit of glycine and serine residues, which will end up being the spacers, and then interspersed along the sequence are a few aromatic residues, particularly phenylalanine and tyrosine. So one of the things that emerged after a lot of work, which I'm summarizing down in one slide, was that the threshold concentration for phase separation under fixed solution conditions is basically inversely proportional to the valence, i.e. the number of the aromatic residues, which are the stickers in this case, squared, of course, because essentially these are intermolecular interactions. And so much so that if at a given set of solution conditions, one were to measure the saturation concentration or threshold concentration for phase separation, the rank order would be this arrow plus variant would have the lowest saturation concentration, followed by the wild type, and followed by these two other variants where we have basically dialed down the aromaticity. So this and a series of systematic investigations basically led us to the conclusion that in these types of systems, the stickers that provide the chemistry-specific associative interactions that drive phase separation are the aromatic moieties, and the glycines and serines are effectively controlling the overall solubility profile in such a way that you essentially can tune the associativity as well as the solubility somewhat independently. What is also fascinating is that an evolutionary analysis reveals to us that these aromatic stickers were effectively evenly distributed along the linear sequence shown here for wild type. And in fact, the likelihood that you would get such a uniform distribution turned out to be something like 10 to the minus 40 if you were to simply do this by random. And so that caused us to ask the following question: what would happen if we were to accumulate these aromatic groups into super stickers along the linear sequence? And in fact, what you find are these sequences precipitate out at very, very low concentrations down in the picomolar ranges, actually, forming a lot of sort of micelles upon micelle type of architectures. And so if the intent is to drive these micron scale puncta that have some of the attributes of liquid-like materials while also showing viscoelastic properties, then the design principle is that one should have aromatic stickers. If one is looking at prion-like domains, these are not the only types of stickers, but that these stickers should be uniformly distributed along the linear sequence. But we've continued to sort of explore the question of what else is going on. It's not just about aromatic residues. One of the things that jumps out, of course, is that the compositions are not as low complexity in nature as one would sort of glibly argue for. It turns out that two things are true. One is that there is a fairly complex composition to these prion-like low complexity domains, but also that this compositional complexity is actually very well conserved across homologs that include orthologs and paralogs. And that caused us, and there are some interesting trends actually. One of the things that we find is that the fraction of aromatic groups is actually fairly tightly conserved, but there's an interesting anti-correlation between the fraction of tyrosines and the fraction of phenylalanines, for example. And so we started to wonder if these were perfectly interoperable with one another, or if there were rules that essentially led to a choice of tyrosines given certain contexts or choices of phenylalanines. So the first thing we do is actually interrogate the energetics of these systems. So one of the things one can do are sort of titrate the sticker types. So effectively, what I'm showing you here are constructs that were designed to change the relative ratios of phenylalanine to tyrosine, two aromatic groups, the only difference being that the tyrosine basically has a hydroxyl group. So the -9F plus 3Y basically is one variant. So essentially, and likewise, we can titrate the tyrosine and phenylalanine content. And what one finds is actually that the threshold concentration for phase separation is temperature dependent. One can perform a Van 't Hoff analysis and actually extract thermodynamic parameters like the enthalpy change associated with transferring these molecules from the dilute phase to the dense phase. The lower these values in terms of sign, because these are negative values, the larger magnitudes imply stronger interactions. And then, of course, the intercept will sort of give us the entropy change associated with this transfer. And essentially what we find is that when we increase the tyrosine content, we end up with very strong interactions. If we basically have no tyrosines, then we have weaker interactions. And so you can actually see now how the energetics starts to dictate the types of both the driving forces for phase separation and also the entropy within the dense phase, if you will. So with these rules in hand, we've been able to, at this point, sort of make measurements, but also do a lot of calculations where we can actually take amino acid compositions and both calculate and or measure saturation concentrations at prescribed solution conditions. What this slide is basically pointing out is that, quite remarkably, in a sequence that is ostensibly intrinsically disordered, and that where we're making really modest numbers of changes, some are fairly large, some are fairly modest, one can actually engineer sequences to have driving forces that are 2 to 3 orders of magnitude apart from one another, right, for phase separation. So what the central message then becomes that when our folded molecules are populating the unfolded state, the interactions that they have among themselves are likely to be reminiscent of what these intrinsically disordered proteins encounter with one another. And these driving forces for phase separation depend quite sensitively on the amino acid composition, is really the central message of what I'm trying to say to this point should be. So some rules that sort of emerge in one category of sequences, not across the board, in one category of sequences, aromatic groups are the primary stickers. Tyrosines and phenylalanines are not equivalent as primary stickers. It turns out that arginines play a fairly special role in that they can sort of either be spacers because of the highly favorable free energies of solvation that they bring to the table, or they can be context-dependent stickers that sort of engage in cation-pi interactions. There's a very, very interesting set of results that has emerged from the phase separation field, demonstrating the complete non-equivalence of arginines and lysines with one another. And in the context of these preon-like low complexity domains, lysines really do act as destabilizers of the interactions between aromatic stickers. And not surprisingly, net charge actually has a very important role to play. Now, I've glossed over all of this because this is all recently published. I want to return to the topic, which I started to discuss at the outset, and that is, how do these rules that I've tried to summarize here without showing you a lot of details map onto thinking about the phase behavior mediated by interactions among unfolded proteins? And that's where we'll dig deep next. This work that I'm going to tell you about is a very fantastic collaboration with the group of Danny Hatters at the University of Melbourne. His student Yunhee Che was a major driver from his side, and Kirsten Ruff, a longstanding colleague in the lab, who has basically been an absolute hero in terms of thinking about these fairly complicated data. So instead of thinking about challenging molecules like superoxide dismutase, where we clearly have oligomerization confounding our thinking, we decided to answer really what was a conceptual question, is how do you link a two-state behavior, a folding-unfolding reaction, to large-scale phase behavior. Barnase is, of course, one of the poster children, if you will, of two-state proteins, has been studied to death by several groups. And Danny's group had shown that a lot of the well-characterized mutations, characterized first in the lab of Alan First, actually have in-cell stabilities that match the in vitro measurements. And what's interesting is that destabilization of barnase in mammalian cells, so this is a bacterial nuclease, which can be rendered inactive by a couple of mutations and therefore can be used in mammalian cells as a probe protein. So destabilization of barnase in certain cells, and these are HeLa cells again, start to form these inclusions that are colored in these red envelopes. And here's a particular mutation. So this led us to the thought that we could use barnase as a model protein to try to uncover some first principles, if you will, about the linkage between the protein folding and unfolding reaction and phase behavior. So this is the topic that we'll touch upon. Now we could do the following, which is, express or transfect a mutant, vary and wait forever, see some inclusions emerge, or we can essentially use some elegant tools developed by the Brangwyn lab to get at essentially reversible phase behavior rather than focusing strictly on irreversible phase behavior, which really would just be a long-term ripening or coarsening of the types of condensates that people are accustomed to seeing. So here, the test protein would be barnase, a fluorescent reporter would be mCherry, Cry2 is an oligomerization domain, and so many of you will start to worry about, all these appendages and what that does to the protein, to the protein of interest. And so independent measures have been made of protein stability with these appendages, and the trend lines basically follow what one sees in vitro for the protein alone. So then the idea is that you activate the oligomerization domain, namely Cry2, with blue light that effectively sets up a way of sort of collecting molecules locally. And then you can ask whether there's something above and beyond the Cry2 induced oligomerization, i.e. does the test protein have the requisite oomph, if you will, to drive phase separation? And so here is the intrinsically disordered region made popular by Julie Foreman-Kay's lab. This is a disordered region from the helicase, dead box helicase DDX4. After light activation, this is basically a 1-minute activation, one starts to see these puncta. One can do interesting sort of morphological and material property characterizations of these puncta. But then when the light is shut off, basically they dissolve over time. And these light bursts are given in sort of 1 to 1.5 minute aliquots because blue light, sustained blue light exposure turns out to be quite phototoxic to cells. If one takes wild type barnase transfected at a particular concentration and carries out this blue light activation, basically what one sees is just dispersed molecules. However, if we have a particular mutant of barnase, in this case a double mutant that destabilizes barnase, one starts to see the appearance of these puncta. The switching off of light will essentially switch off these puncta as well. And that's shown here. So basically we have before activation, this is at a particular concentration or a particular level of transfection. Then we turn on the blue light. You see at different time points after activation, and then essentially here the light is off, and then you see the dissolution of these puncta. So these are what are called optodroplets. And basically the question now is the formation of these optodroplets sensitive to the concentration of the protein, in this case, the Barnase mutant? And is there a threshold concentration above which one sees the appearance of these optodroplets? And how does that threshold concentration depend on protein stability? So basically, what one observes is that there are variants that basically are destabilized marginally with respect to the wild type, but they never really form puncta, no matter the expression or transfection level. And then there are a series of mutants for which there is a mutant-specific threshold concentration above which one can start to see the appearance of these light-activated puncta. So after scrutinizing a lot of data, Kirsten came up with a really simple model that basically says the following, that there is a total concentration that has to exceed a certain threshold we refer to as the saturation concentration. But in addition, there is a contribution from protein stability, which sets the fraction of unfolded molecules, which we call Pu. And the product of these two quantities leads us to a threshold concentration of unfolded proteins, which when crossed gives rise to the appearance of puncta. So this model was forged by the analysis of about 10 different mutations. And this is essentially at this point a hypothesis. So let's go back and ask what that does for us. So essentially, the crossing of C star, so CSAT essentially is, the total concentration of Barnase. The crossing of this threshold concentration of unfolded molecules, of course, is now set by the protein stability. And so if the free energy of unfolding is highly favorable, that means pretty much all the molecules are going to be unfolded, then C star essentially is immeasurably small. But as the stability increases, one can start to see that essentially the total concentration of Barnase molecules required to cross C star is actually quite large, right? So essentially the C star is a fixed number. CSAT in this case is the one that's varying because essentially there's a linkage between folding, unfolding, and phase separation. So with that in hand, we were able to, we being Kirsten here, arrive at a model that basically looks like a phase diagram where we have total Barnase concentration along the x-axis, free energy of unfolding along the y-axis. And here is this sort of phase boundary that basically says that when this boundary is crossed, we start to see phase separation. So essentially the idea is that a threshold concentration of C star, that happens to be some arbitrary value in terms of fluorescence units, is required for driving phase separation. And this will be set by protein stability is the model that emerges. And so one can go back and ask the following, which is, to the left of this is where we'll see phase separation, to the right is where we won't. And so we can actually start to see what sort of phase behavior we actually obtain. And it turns out quite a few mutations sort of fall within this threshold, sort of satisfy this threshold concentration for phase separation. But there are lots of others that actually don't, right? So there are outliers in terms of C star. So remember what I said is that the concentration of unfolded molecules should cross a certain threshold C star, and that's what that is set here. And so what we can ask is, is it the case that every mutant that sort of crosses this threshold concentration in terms of C star undergoes phase separation? But the answer is no, not quite, right? And so what goes on? So it turns out that phase separation is clearly governed by more than just protein stability, which sets C star. There must be something else to this, right? So the hypothesis that then emerged was that if mutations end up deleting stickers that drive unfolding, then the unfolded proteins will, of course, remain soluble. They don't have to, because essentially you've destabilized the protein so much that there is no driving force for folding. So the stickers are lost, which means that such stickers now don't exist to drive intermolecular associations and phase separation. So unfolding alone is not a is a necessary condition, but not sufficient condition. So that then led to a very interesting hypothesis, which is that there must be stickers then that do not contribute to the stability of the folded state, but must contribute to the association of unfolded molecules, right? So this led us to sort of exploring this question of trying to identify the stickers that drive phase separation of unfolded molecules. And the way we did this was using a simulation approach that we'd sort of developed over the years in collaboration with Dan Raleigh's group, which basically said we have a way of understanding what unfolded states under folding conditions will actually look like. They typically look like Flory random coils. And based on these simulations, we can identify stickers that form contacts. So these would be non-native contacts only in the unfolded ensembles. So I want to be very, very clear here. What we're saying is that the stickers that drive intermolecular associations of unfolded molecules must make contacts only in the unfolded state, not in the folded state. And it turns out that there are quite a few of these given the heteropolymeric nature. So these are basically the small circles that have hot colors here. And lo and behold, we're talking about residues that fit exactly the sticker grammar that you would have, that we uncovered from looking at IDPs, quite a few aromatics, and of course, there are quite a few aliphatic hydrophobic groups as well, but the dominant entities are the aromatic groups, right? So this started to suggest to us that, hey, we could do the following. We could make mutations that basically only impact the interactions of unfolded molecules with one another and then query what happens. So we did that. So here is an 8-ALA mutant that essentially is effectively 100% unfolded. You can stimulate it with blue light. You can start to see the formation of puncta. But take that 8-ALA mutant and replace the stickers with serines, which we had shown before are actually bona fide spacers. You basically lose the ability to activate the formation of puncta. But then if we start adding more stickers, right? So effectively we now, we do two things actually. We either add more stickers, so these are glutamines substituted with tyrosines, or glycines substituted with tyrosines, or threonines substituted with tyrosine. Or we change the identity of the aromatic sticker, because remember what we discovered is that, phenylalanines and tyrosines are non-equivalent. We basically start to see sort of robust puncta formation. But again, these are just qualitative images. Since we do these measurements at a series of concentrations, one can actually extract the threshold, essentially the concentration of the mutant at which you see phase separation versus what you would see for the default unfolded, highly unfolded construct, which is the 8-ALA construct. Any negative numbers means that the driving force for phase separation has been enhanced. And a positive number would mean that the driving force for phase separation has been suppressed. And essentially what's up here is we don't observe phase separation at all across a concentration titration that goes about almost 3 orders of magnitude. And what we essentially find is that replacing stickers with spacers, so the stickers here are now the wild type ones that I showed you on the earlier slide, with spacers basically results in no phase behavior. The substitution of replacement of phenylalanines with tyrosines tends to have only a modest effect. It's not as strong an effect in the context of these molecules as it was in the context of the prion-like low complexity domains. And we understand that now because in work we've done with Tanya, we know that there's an interesting covariation of the sticker types with spacer types that actually gives rise to the differences between phenylalanines and tyrosines. So it's not just intrinsic energetics, but cranking up the number of stickers now, where we replace spacers with stickers, clearly increases the driving forces for phase separation. So what this now says to us is that phase separation via interactions between unfolded molecules requires both a requisite stability threshold to be crossed to set the concentrations of unfolded molecules to be above some threshold, and the presence of the appropriate set of stickers that give you chemistry-specific interactions. I don't say site-specific, because it may well be that they're site-specific if particular structures are also needed, but they're certainly chemistry-specific. We cannot, with our resolution, say anything about whether these are forming specific structures. What we do know is, using blunt-edged probes, they don't happen to be amyloidogenic. So effectively then, what we've emerged with is an interesting result, I think, which is that just being unfolded is not a terrible thing, but a necessary condition for phase separation of stable proteins is that they be unfolded, or the concentration of unfolded molecules cross a threshold C star. That C star is determined by a combination of stability and, of course, total expression level. So it's not just one, but it's actually both. And for unfolded molecules to sort of drive the formation of some kind of agglomerate, be it a punctum, an inclusion, a liquid, we're agnostic about what that is because we're not focusing on the characterization of that end just yet. What we need is a requisite valence of stickers, but most importantly, these are stickers that basically form contacts in unfolded states, which some people think of as gatekeeper interactions in sort of controlling protein folding reactions. But in this particular case, we're essentially talking about stickers that are, from the context of the folded state, making non-native contacts in the unfolded state. So this leads us to the question then of, okay, so if there are some general principles, then how do components of the proteostasis machinery respond to the presence of puncta driven by the association of unfolded molecules? Remember again, here's how we define C star. And so the observation is that the overexpression of HSP40 or HSP70 or HSP40 and 70 generally increases CSAT, which means that the total concentration needed in order to achieve C star is actually increasing. So effectively what's happening is a model that will emerge shortly, is that these chaperone molecules are preferentially interacting with the unfolded states. Of course, there is location-dependent behavior here, where basically what we find is that the effect of these chaperones is either nucleus-specific or cytosol-specific, and it's tied to the abundance of these proteins in these relative compartments. But HSP40 is you look at the HSP40 effect, it's actually quite a bit more significant than the HSP70 effect. And so this leads us to effectively a model that looks like this, right? It says that chaperones are actually affecting phase boundaries, meaning that by preferentially interacting with the molecules that can drive phase separation, the underlying threshold concentration for phase separation, or the concentration that one needs to cross, is actually shifting upward. So it turns out that this is a well-established concept in the literature, well-established but long forgotten, the concept of polyphasic linkage, whereby one can regulate the driving forces for phase separation via ligand binding. And this was introduced first by Wyman and Gill. We rediscovered this, and we've been able to sort of uncover a series of rules that sort of help us understand this ligand-mediated phase behavior. The physics goes quite simply as follows. When we see two-phase behavior, we have a macromolecule shown here in red that is essentially partitioning into two phases. We'll call them A and B. And the phase boundary is set up at a concentration that is dictated by the equalization of the chemical potentials. So what we end up with is a concentration of macromolecules in the dense phase labeled B and the dilute phase labeled A. And these concentrations are set by the equalization of chemical potentials, which of course are nothing but partial molar free energies. Now we add a ligand, right? And the idea is the ligand doesn't have any interesting phase behavior of its own, but essentially we now have a new threshold concentration for phase behavior in the presence of ligand, and that will be governed by the product of the intrinsic saturation concentration and the ratio of binding polynomials, where our ligand essentially now has, excuse me, differential affinity for the macromolecule in phase A versus phase B. If binding is equivalent, then the driving forces for phase separation are not impacted. But if binding is non-equivalent, we can get some interesting behaviors. So here, we'll talk about a macromolecule or a scaffold whereby the ligands preferentially bind to the macromolecules in the dilute phase. Here's the phase boundary that sets up these two coexisting concentrations. Because the ligand likes these macromolecules in the dilute phase, the threshold concentration for phase separation is shifted upward. And in fact, even the dense phase is now diluted. So essentially the ligand is spigoting molecules out of the phase separation reaction through preferential binding. And so I want to point out that this is very different from partitioning, right? This is basically saying that the ligand can interact with the molecule, macromolecule, in both the dilute and the dense phases. But either the type of binding, the overall affinity for binding, the cooperativity of binding, that is different across the phase boundary. And that difference sort of leads to the exertion of ligand-dependent phase equilibrium. We can have the opposite scenario, which is that there maybe are more sites in the dense phase, or there's cooperativity of binding in the dense phase, in which case now the ligand will essentially enhance the driving force for phase separation. That means the threshold concentration for phase separation will actually go down when compared to the ligand-free case. And so now you can think about components of the proteostasis machinery, at least the so-called energy-independent entities, really as ligands that are modulating phase behavior. So I'll show this to you using Huntington, because we actually have, because this example actually sets up what one should measure in the context of getting at ligand-dependent phase equilibrium. This is the work of Ammon Posey, a senior scientist in the lab. So here's a reminder quickly of the types of constructs we work with in the lab. Here is an N-terminal exon 1 spanning fragment of Huntington. We titrate the poly-Q length. I'll show you results for a poly-Q length of 40. There is, of course, this N17 construct that is essentially a helix-forming entity. And there's a proline-rich stretch that is going to become quite important because of these two polyproline modules. So over the years, what we've shown is that Huntington actually shows complex phase behavior on its own. It's a molecule that actually has some striking resemblance to surfactant types of molecules with this tadpole-like architecture that we uncovered in collaboration with Hilal Lashual and Edward Lemke. This is essentially at this point the sort of highest resolution structure, if you will, of the exon 1 spanning fragment of Huntington in isolation. We showed that these molecules can form these interesting micellar phases. We call them spheroidal phases. They're fairly monodisperse, actually, at fairly low concentrations. In the threshold concentrations about 100 nanomolar. These threshold concentrations, as opposed to specific structures, are what change with poly-Q length. And then there's a threshold concentration above which these fibrillar phases form, and these are sort of rod-like structures. And so here's the three phases you can think of: a monomer or oligomer phase, a spheroidal phase, and a fibrillar phase, accessible in different concentration regimes. So let's go back and think about a ligand that happens to be profilin, which Mark Diamond's lab showed when overexpressed will actually lead to a reduction of both aggregation and associated cytotoxicity, both in cultured cells and also an animal model. And in fact, this idea of targeting the phosphorylation of profilin is one of the early drugs known as Fasudil that is still marketed in Japan for treatment of Huntington's disease. So profilin is a small 15 kilodalton protein that actually has a lot of interesting things going on with it. It can bind G-actin, it can bind PIP2 across its surface, but it can also bind polyproline. And you will well remember that Huntington has 2 polyproline modules. We discovered that this binding of polyproline modules in Huntington to profilin is actually independent, but yet profilin exerts a fairly interesting effect on the phase behavior of Huntington. So I'll show you measurements of how we quantified this using the impact of profilin on the S phase. So the idea here would be that here is the intrinsic equilibrium defined by a threshold concentration of a few hundred nanomolar for the formation of the S phase for a given polyQ length. If profilin were to bind preferentially to the M phase, meaning predominantly monomers and small oligomers, then the threshold concentration should shift up, right? Conversely, if profilin were to bind preferentially to the S phase, because there are some interesting effects potentially available to profilin, nonlinear effects in the context of the S phase, then you should see the threshold concentration shifting down. So Ammon actually spent quite a bit of time and then came up with a very nice assay that's based on a simple cuvette-based measurement of these threshold concentrations. And in fact, what you can do is titrate the concentration of Huntington, fix the concentration of profilin, and measure the threshold concentration for the formation of the S phase. And what you find is that in a profilin concentration dependent manner, this threshold concentration shifts up by well over an order of magnitude, depending on the amount of profilin there is, right? So this basically is suggesting that what we're observing is preferential binding of profilin to Huntington molecules in the dilute phase or in the M phase, which shifts the concentration upwards. So what emerges is a model that looks like this. Here we've got our 3 phases. In the presence of profilin, what we start to see is a shifting of the phase boundaries upward, so much so that you can actually crank up the concentration of Huntington molecules to be in the hundreds of micromolar range, even for pathological lengths. And you don't really see fibrils forming. And that has entirely to do with the preferential binding of profilin to the M phase. Again, a reminder, this does not mean that profilin does not bind to these dense phases. It actually does just fine, but you would essentially mislead yourself if all you did was measure the binding of profilin to the dense phases, because one wouldn't know what is actually going on with the underlying phase equilibrium. And in fact, we now have quite a bit of data showing that the model that sort of best explains these data, or that is the one where a single profilin binds to the polyproline, but also the G-actin binding sites are able to engage in considerably weaker interactions with the polyQ domain. So this multivalence actually engenders this type of linkage phenomenon. So the key point then becomes that one can use, in the case of Huntington, in the Huntington exon 1, we don't really have any conformational changes we were necessarily thinking about, because even if they're there, they're far too subtle for us to be able to measure. But essentially binding across phase boundaries basically sets up these linkage reactions whereby one can actually think about how ligands mediate the regulation of protein homeostasis. So effectively then the model that comes about for ligand-mediated phase behavior would be that things like chaperones, ATP-independent chaperones, would essentially do so through these preferential binding reactions. And essentially what we have is a folding-unfolding equilibrium and a phase equilibrium. They are linked through the concentration of the unfolded molecules, but also through a grammar that effectively dictates whether or not these unfolded molecules can stick to one another in a specific way to generate particular types of phases. Now, in the presence of a ligand, essentially now what we do is we renormalize the concentration of unfolded molecules by binding preferentially to them. And again, I think we can go well beyond just saying, you know, this is generic exposure of hydrophobic groups and so on, because the same interactions that are driving the phase separation are also enabling the interactions with the ligands, but there could be other interactions as well. So the key takeaway I want to sort of leave you with is that if a molecule is happy being unfolded and is incapable of chemistry-specific or site-specific or structure-specific interactions, I rather suspect these are unfolded molecules that if they come in the way will just be degraded, but don't really cause a lot of problems or emergent phenomena for cells. It's a brave hypothesis, but one that I think is supported by a lot of emerging data. So let me stop there. I think I've some time for questions and answers, and I will be happy to take any questions you might have. Thank you.
Great, thanks very much, Rohit. I see we have one question so far. So if anyone does have questions, please type them into the Q&A and then I'll read them. So our first question is, what about tryptophan? It's a hydrophobic aromatic residue. I realize it's less abundant than Phi or tire, but it may have a special function in pinning because it partitions to the interface.
Yeah, sure. I mean, you know, I think this is a question that comes up a lot about tryptophan, and certainly it is. I mean, we have data showing that it is a way more strong sticker than tyrosine or phenylalanine. Doesn't really show up as much in a lot of different proteins. And you use the word pinning, which of course has a very specific meaning, and I'm not sure if that's the meaning that you intend, but the follow-up sort of suggested that that is the intent that you have. So it's not clear if the interface that you're referring to is the interface in terms of the phase boundary. We don't know that. I think what you're referring to here with regard to tryptophan has a lot to do with how it partitions to the interface between, let's say, membranes and the aqueous environment. This leads us to an interesting question of, you know, is the inside of a condensate sort of akin to the inside of a membrane? But, you know, that is not something that is a resolved issue at this point, and I suspect is also going to be very sequence specific again. But yes, if if tryptophan were present, it would be a very potent sticker. Absolutely.
Thanks very much. Our next question is, a ligand could also enhance aggregation or decrease the critical concentration for LLPS, right?
Yes, absolutely.
Okay. Our next question, and from your talk, I understood that the unfolded state is a necessary condition for phase separation. Also, you mentioned some sticker interactions specifically work in the unfolded state. So I assume in the phase-separated entity, a significant population of the protein is in the unfolded state to maintain those sticker interactions.
That's an interesting question, okay? Because what sort of conformational transitions are engendered by the sort of unique environment of a dense phase is to be interrogated in the context of the dense phase, right? And this is something that is what your question or your comment sort of touches upon is something very, very important. All of us, and I mean all of us universally, are very good at measuring interactions in dilute phases and extrapolating what that means for, what happens across a phase boundary. But these are emergent phenomena, right? So just to give you a very specific example, we published a paper late last year showing that glycine-rich tracts, which, by the way, in our parlance for intrinsically disordered proteins would be predominantly spacer residues. They, at the types of concentrations you will see inside of dense phases, make beautiful fibrils, right? We're not talking about hydrophobic groups. These are really polyglycine tracts. And in fact, the uninterruptedness of a polyglycine tract turns out to be quite crucial for the formation of these types of fibrillar architectures. So what that means then is that there is an emergent behavior that potentially awaits some of these molecules, and which is why I think the processing of these dense phases or the expenditure of energy to sort of maintain fluidity is a theme that you see oft quoted in the literature, because it's not clear that the interactions have to be identical in the dense phase.
And then let's see, spherulites, also micrometer spherulites are normally always made by unfolded proteins. Question.
Yeah, that's a perfect question that segues directly off what I just said, which is that, yes, you can get these types of spherulites from inside of dense phases or potentially from interphases, but you're absolutely right. Typically, what they will require are interactions among unfolded molecules that are pinned down somewhere, right? So.
Okay. And then let's see, Sheena comments a wonderful talk, which I echo. And then she comments, when you talk about unfolded states being highly disordered, could partially structured states play a role in phase separation? And hence the stickers and spacers might need to be correctly organized in three-dimensional space as well.
Right. So this is a really important point, and I apologize for glossing over the details of the model that we constructed for the unfolded state. I just assume that everybody here reads every paper that Dan Raleigh and I write together. So
well, I do.
So the clarification, Sheena, is that the model for the unfolded state is not the one that you access in, let's say, 8 molar urea or 6 molar guanidine. It really is the type of unfolded state that you would access under folding conditions, right? And so under those conditions, essentially what you see is, as you indicate, a lot of partial structuring. And so in Kirsten's models for the unfolded state, these non-native contacts emerge from particular features of the unfolded state. We don't focus on the specific structural motifs because there's a kind of redundancy in the types of structural motifs that can give rise to these statistical interactions, because they don't always have to be precisely one structural motif. But if that is indeed the case, as is true in beta 2M, then you're absolutely right. The sticker now is made up not just of chemistry, but as I hinted in a few times during the talk, also by the motif, the structural motif that is adopted.
Perhaps I could ask a follow-up question before we go to the other ones that are posted. Hey, do you have any thoughts on the stickers balancing intermolecular interactions and intramolecular interactions in the folded state. One may inhibit aggregation, the other might promote it. Is there?
Yeah, so that's a really important question, where I think the way to think about it is we're looking now at a competition between a unimolecular reaction, i.e. folding. So I think what you're hinting at is perhaps non-native interactions that are gatekeepers, that perhaps don't, that in some ways inhibit promiscuous intermolecular interactions. So that's absolutely the case, right? And so one of the ways to think about this is that in ultra dilute concentrations, the likelihood that you will actually have competing intermolecular interactions is very, very low. The idea, that's why there is this threshold concentration, right? So effectively, the idea is that above this threshold concentration, the overlap of molecules, or in fact, the likelihood that molecules see one another through intermolecular contacts, is going to be higher. And so now you have essentially a competition, and that competition sort of tilts in favor of the intermolecular associations as the concentration increases. And so the simple answer to your question is that competition is entirely predicated on the total concentration and the presence of a threshold for being able to drive associations, intermolecular associations versus only intra.
Okay, thank you. Let's see, we're getting quite a few questions now, so let's see how many we can get through. There's a question about, could you comment on the role of common amyloid cofactors, such as glycosaminoglycans, lipids, metal ions, et cetera, in the context of phase separation of unfolded proteins?
Well, so I can offer a very generic comment, and that would be, and the protons is a whole other ballgame, right? Because now we are in sort of, this is where I think we can wax eloquent for at least a few hours, because this heads us back into the types of things that both Dan and Sheena have studied over the years, the notion that you get very strong pH dependent effects. And here it really would be site-specific modifications, right, that are sort of impacting the underlying balance of interactions and so on. Life gets a little more tricky with things like ions, but again, I would say the underlying framework should be more or less the same as the polyphasic linkage ideas that Wyman and Gill developed, which is, how do these cofactors impact the molecules, or do they preferentially interact with these molecules in one phase over the other? Again, that doesn't come about because it doesn't interact or doesn't partition with a particular phase. That's an extreme example. But given sort of competing interactions, is there a preference for one phase or the other? If that is the case, then you will see all of these cofactors sort of having an impact on the underlying phase behavior.
Based on the role of the aromatic residues that you propose, do you think it would be possible to have liquid-liquid phase separation without any aromatic residues?
Oh, absolutely. So in fact, I think I tried to caution myself as I went along. I extracted rules primarily from the preon-like low complexity domains. So the way we're thinking about these sequences that drive phase separation, there really is a basis set of sequences. We're pretty good at sort of understanding now what happens with these preon-like domains. They have been sort of among the most popular ones, but directly to your point, even in the full-length proteins, it's not just the preon-like domain that's doing much of the driving of phase separation. It really is the synergy between the preon-like domains and the RNA binding domains. And in this particular case, it looks like we have a sort of network of arginine, tyrosine, arginine, phenylalanine interactions that really is important. But also, in the context of the nucleolus, in the context of speckles, just to, in fact, give you a very tantalizing sort of observation, we've discovered that RD repeats right? So arginine aspartate repeats make these stereolytic structures that were alluded to earlier. There are no hydrophobics in there. There are no aromatics in there, right? Whereas KD repeats won't do such things. In the context of the nucleolus, it really appears that it has nothing to do with aromatic moieties per se, but it really is sort of the linear segregation of oppositely charged residues inside of these sequences that drives phase separation. So we fixated on the aromatics for this particular story, and actually hydrophobic groups, mainly because the way we were thinking about it was that, okay, there are driving forces that stabilize folded states. You destabilize a folded molecule by sort of weakening those interactions. But then we realized, oh, wait a minute, you'll populate the unfolded states quite efficaciously, but you may not end up, you might not have the stickers to drive phase separation. But of course, that's a statement that's only true if the contribution of charged residues and polar groups is weak in the context of these unfolded molecules, which appears to be the case, but that needn't be general.
Okay, great. So I realize a couple people have asked follow-up questions to their earlier questions. I'll return to those, but I'd like to try and get through the first set of questions if we could, so everyone gets a chance. You mentioned phase separation will be governed by chemistry-specific interactions. And the question asks, is it possible that these chemical specific interactions can occur without any specific amino acid sequence?
Yeah, that's the general idea when you think about sort of intrinsically disordered proteins. I mean, when you're talking about conformational heterogeneity, what we're essentially saying is that, you know, I can sort of arrange for networks so long as I have the appropriate composition. But again, I want to point out that if I have come across as making sweeping generalizations, then I have seriously erred in my presentation, because what I said today sort of applies in the context of unfolded molecules being conformationally heterogeneous and so forth. What we do know is that there are lots of stereospecific interactions that do drive phase behavior. The key question is, what does conformational heterogeneity buy you? And that it buys us two things, right? One is it buys us multivalence and therefore promiscuity of interactions. But promiscuity of specific interactions, if that's a usage that I'm allowed. And the second is that this conformational heterogeneity allows for molecules to overlap with one another at really low concentrations. And so the first order determinant will always be composition, but you can always, as I showed you, we can repattern the stickers versus spacer linear sequence features and basically drive precipitation at ridiculously low concentrations, right? So clearly the details of the specific amino acid sequences also matter. And I will caution that I think, I never thought I'd sort of wax eloquent about, you know, in my time I've seen this, but, you know, I'm old enough, I can say this. I think too often we tempt ourselves into sort of making these generalizations, and invariably there will be a use case in biology that will stump us. And so I think, you know, a certain humility is important here in terms of, you know, learning from the systems as we uncover the underlying rules.
The next question, I think you largely addressed. It was about the importance of heterogeneity in the unfolded state and how dependent is this concept on conformation. But perhaps the one part of the question that you could speak a word or two to is how can one since whether one is a sticker or a spacer, part depends upon case. How could one determine a priori which are stickers and which are spacers before one does measurements or makes mutations? Is that possible in the current framework?
Absolutely. So in fact, one of the nice things that emerged from Kirsten's simulations was really sort of a prescription for how to identify stickers, right? Her prescription was actually completely agnostic of the stickers being aromatics or charges or anything. Basically what she said, and again, this is where I want to emphasize that roughly 2+ decades of work in several labs, Dan included, Sheena Radford included, Julie Foreman-Kay included, really focusing on describing unfolded states under folding conditions has turned out to be immensely profitable for our way of thinking, because through that sort of close connection with experiments, we've developed sort of a way to describe unfolded states at atomic level resolutions, requiring that under folding conditions, these sort of adopt, an effective property of being like they're in a theta solvent. That's sort of a requirement. But essentially what that allows us then to do is to go back and say, if I know the structure of the folded state, which of course, thanks to AlphaFold, you at least have a prediction of, then what you can do is go back and say, from the simulations of the unfolded state, you can go back and ask, what are the regions or residues or structural motifs that are preferentially interacting in the unfolded state, but not so in the folded state? And that becomes your first order way of dissecting stickers. Sadly, that requires that you do those computations, and they're not necessarily trivially expensive. So there is no server that I can sort of offer you and say, go put this up here. But I'm hoping that as we do more such simulations, we can sort of turn to a machine learning paradigm that will allow us to sort of move in the direction of a server, but we're a ways away from that.
Okay. Our next question is, has anyone done field theoretic simulations of these systems? If so, do they reproduce the threshold concentrations in free energies that you see?
Yeah, so in fact, Joan Shea is here. Joan has worked very closely with Glenn Fredrickson over the years. Glenn, of course, is sort of very well known for developing these field theoretic approaches. And so, yes, they do work. Joan has actually been remarkably successful in sort of prototyping these for certain model peptides. To the best of my recollection, and Joan, please correct me if I'm wrong, that those brand of calculations have not been adapted to the types of problems I discussed today, but they have been applied to sort of polyampholytic systems and polyelectrolytic systems where the physics of complex coacervation, so charge-mediated interactions, become very relevant and important. And Joan has actually published at least 3 papers that I can recall off the top of my head, possibly more.
Thank you. Let's see. In unfolded states, the protein could maintain its local secondary structure, ?
Yes. Statistically, yes, absolutely.
And then when the stickers or other mutations are introduced in folded states, this is, I believe, speaking to the context of your model, how does this affect the population of the unfolded state? Or is the unfolded state population assumed to be constant in the model?
Yeah, so I believe what Grace is referring to is the part where we actually introduce de novo mutations, not just destabilizing mutations. So sorry if I wasn't clear, all those sticker insertions or spacer titrations were done on the background of this 8-ALA mutant, where essentially we already know that the unfolded state is pretty much 99% of the population. Right, so there is no, this is independently verified to ensure that we don't really observe any folding. So in the way we calculate C star, we already know that C star is set now, right? Because essentially that's prescribed by the fact that we have 8 mutations, 8 residues in RNAs turned into alanine that have essentially massively destabilized the protein. And so, yes, and so in that particular context, the unfolded state population is fixed. But to arrive at the model, it ended up using quite a few different mutations, where the population of the unfolded state was also titrated.
Okay. Thank you. Then Carmelo asks, thanks for the interesting talk. You opened about symmetry breaking. Please explain to me what it is, as I didn't understand well. You talk about stickers in aggregation without considering the secondary structure of the polypeptide chain. At the same time, the symmetry breaking hypothesis is based on the secondary structures of two monomers that aggregate to give a dimer or a trimer, et cetera.
Yeah, so just to clarify, I believe you're referring to slide number 2 or 3 rather, where we were, I was very clear in pointing out that the concept of symmetry breaking was in the context of amyloid formation, which is really about the formation of de novo secondary structures that can then sort of grow into some kind of a semicrystalline state. We do not have evidence for amyloid formation in these systems, and therefore we do not have evidence for the symmetry breaking. So yes, your understanding of symmetry breaking is absolutely on point. I just don't discuss it because we don't have any evidence for that.
Our next question, I appreciate your attempts at the abstraction to stickers and spacers. Ultimately, the interactions of heteropolymers are to be understood from the energetics. Jeffrey Wyman is a hugely important reference. Do you see your abstraction at variance with this view? When ARG may be context dependent, and maybe many other things can be as well, such that we have to go down into the details.
I'm not entirely sure I understand the question. I think there's a criticism of some sort being leveled here, but I'm not being able to put my finger on the criticism. So yes, we have put numbers on the energetics, right? So essentially, what I talked about was we can actually rank order stickers in terms of their strengths. We can rank order spacers in terms of their contributions to the Flory-Chi parameter. We can be fairly confident about the identities of stickers versus spacers in the context of intrinsically disordered proteins. Of course, others, the identities of stickers versus spacers and the energetics of stickers versus spacers will be context dependent. Do we have those rules yet? No. Are we getting there? Absolutely, yes.
Great, thank you. And if that didn't address the question that was posed, please feel free to post a follow-up.
Yes.
I shouldn't speak for you, I apologize, Rohit.
I'm inferring that you didn't think I answered the question, but that's okay.
Louise A's asks, fantastic talk. Wonder if you can comment on the role of RNA in liquid-liquid phase separation in stress granules, and whether one might predict that aromatics are important in the interactions, or if you suggest another part of the molecule might be important, phosphate backbone, et cetera.
Absolutely. So we've published a couple of papers on this topic, one of which clearly shows that there are differences between, so from the RNA side, purines make for better stickers than pyrimidines. So essentially double rings makes perfect sense, right? I mean, so they prefer to engage with arginines over lysines. So even though you would expect cation pi types of interactions, the arginines carry a lot more oomph in terms of their ability to interact with the purines. Purine tracts make for super stickers. And as far as the aromatics are concerned, again, the rules sort of follow the same trends. But I would say in that particular context, so in the context of protein RNA interactions, and this might be a more satisfactory answer to Franz's question, the arginines are way better at being stickers that drive heterotypic interactions when compared to the aromatic groups. But the aromatic groups themselves follow the hierarchy of tryptophan being better. So AGO1, AGO2 are sequences where you get runs of tryptophans that do precisely what you would intuitively expect, tyrosines being then superior to phenylalanines and so forth. But really, it's the arginines that turn out to be sort of the business end of the protein RNA interaction driving thing.
Let's see. As you propose that both chaperones or cofactors could have preferential binding in the concentrated or dilute phase, could this concept be extended to small molecules? Do you think it would be possible to find a drug candidate which functions as chaperones and cofactors do in this context? And could one speculate on the difficulties of doing so?
Absolutely. So I call your attention to a paper that Kirsten Furkan and I published last year, just about a year ago in PNAS, basically working out the rules of what it takes to design ligands that will have precisely the effect that you want, as in preferentially bind the dilute phase or preferentially bind the dense phase, or change the internal organization of molecules within dense phases. And what emerges is actually very interesting. And that is that it, of course, is tied to the energetics, but also more importantly, it's tied to the valence, right? So if one can design multivalent ligands that essentially either bind, so multivalent ligands that bind only to stickers, right? So that's, in that sense, you're already incorporating a site specificity. Then what you get is essentially a preferential binding to the dilute phase because you're competing with the cross-linking interactions that drive phase separation. If, on the other hand, you have multivalent ligands that can bind equivalently with the dense, with stickers and spacers, now you actually are adding cross-links into the system, and you can actually preferentially stabilize the dense phase. And I should point out that those rules have recently been proven to be quite effective in the hands of Carlos Castañeda and Tao Dui, who show that you can model these polyphasic linkage effects, not model really in their case, you can measure them by quantifying the effects of monoubiquitin versus polyubiquitin chains on the phase behavior of ubiquilin, which is an ubiquitin-like protein that also binds ubiquitin. And this is a proteinopathy-involved protein, and the rules are quite striking in terms of their ability to sort of explain the data, right? So the answer to your question is that, yes, I think the rules are starting to, not just starting to emerge, have emerged. And this is actually driving the way people are thinking about bi- and multifunctional molecules. And by that, I mean molecules that have multiple functional groups that can sort of affect phase behavior from the vantage point of the stickers and spacers language. And in that context, I would point to, some early work by George Whitesides and company showing that this is a way in which you think about molecules that impact phase behavior.
Let's see. Very nice and clear talk. Can you comment on the potential physiological role of aggregation of RNA binding proteins, e.g. in short or long-term memory?
Yeah, so this is a sort of very interesting question. Kaushik-C has this very intriguing hypothesis that, more liquid-like RNP granules might play a role in short-term memory, whereas more amyloid-based condensates or phases might have a role to play in long-term memory. It's an interesting sort of hypothesis, I am hardly qualified to comment on it other than saying it's interesting, but I can say one thing, which is that, you can certainly engineer and control hysteretic behavior in terms of responsive phase transitions, right? And so by that, what I mean is that we have historically thought of a lot of these aggregates and sort of phase separation as a genuinely irreversible phenomenon. And I think that sort of has led us astray to some extent, because it turns out that you can always find stimuli that make many of these phase transitions be responsive. But what's fascinating about that responsive phase behavior is the recovery of, let's say, the original phase when you sort of take away the stimulus often shows hysteretic behavior. And so when you see hysteresis, that always gives you the prospect of engineering memory into a responsive system, right? And so that will be the extent of my question in terms of memory. But on the other hand, in terms of other physiological roles, I can tell you that we have a biochemical reconstitution of the nucleolus. And we recently put out a manuscript in collaboration with Marina Ferrick and Tom Mistelli showing a biochemical reconstitution of a mitochondrial condensate, where these are transcriptional bodies in the case of the mitochondrial condensate. And in the context of the nucleolus, it really is an assembly line where you're transcribing the ribosomal DNA, generating a premature ribosomal RNA that has to be processed in one layer, and that comes out and sort of assembles with an influx of ribosomal proteins to generate ribosomal subunits. And so the nucleolus, I'd say, is a striking example of a multi-layered, multifunctional sort of microreactor, right? And so I won't necessarily dwell on memory because that's well outside of my expertise, but I think in the context of the nucleolus, and certainly in the context of nuclear speckles, you can see a lot of very important functionalities driven by these assemblies.
And then I'll come back to an earlier question, which is, so in cells, many proteins are now known to phase separate and form physiologically relevant functions. How do unfolded proteins perform these native functions in the cellular context?
Well, so I hope I didn't convey to you that these unfolded proteins are in fact forming physiologically relevant condensates from a sort of biochemical reaction standpoint, but I suspect the relevant physiological function here is in the context of protein quality control. And therefore, the off-axis would be in the context of disease. So one data set that I didn't go over was we did go back to now think about the compositional specificity of these condensates that are formed by unfolded proteins. And so in a fairly substantial proteomic analysis, what emerges is not only do the components of the protein quality control machinery sort of respond to the formation of condensates or bodies by unfolded proteins, but in fact, it turns out that proteins that have overlapping chemistries, and this brings us to the chemical specificity idea. I think a lot of people fixate on structural specificity, but chemical specificity is such that you now start to see, depending on the type of stickers that are prevalent in the different RNase molecules, the composition of those condensates is governed by the sticker grammar. Right? So you can start to see speckle proteins in certain condensates. You can see ER proteins in certain other condensates. And so the answer to your question would be as follows. I think the formation of these depots or condensates allows for spatial regulation of protein homeostasis, right? Because this is not, protein homeostatic components aren't necessarily, I don't think, dealing with components that are just freely diffusing around in the cell. The ability of these molecules to sort of form these puncta, I think, is important for sort of binding mediated protein homeostatic regulation. But there's the off axis as well, which is the idea that you end up potentially gaining new functions that are potentially deleterious in terms of these condensates sequestering essential components into them. So I think this is this continued battle for sort of life, right? And sort of dealing with this. And that's, in fact, I'd say the condensate formation is an appropriate response because the interactions of the sticky molecules have engendered that, and the cell has cofactors and components there to deal with the problem. But you can also pick up essential components as part of it, and that could be a problem.
Thank you. If there are no more questions, perhaps I could ask the last one, if that's allowed. So I was intrigued in the initial part of your talk with the thermodynamic analysis. The Van Hoff plots were remarkably linear. Does that mean that there's no heat capacity change involved in any of this? And does that speak to the type of interactions that could be playing a role?
That's definitely a possibility. Now, you should remember, of course, that we're analyzing the Van Hoff analysis is performed by looking at the linearity only of the CSAT, right?
Okay.
Not right. So but the C dense doesn't change much, but it's also an insanely noisy quantity to measure. So we don't have as good measurements of the concentration in the dense phase, right? And so we fixated on the CSAT for two reasons, mainly because it's the quantity that varies substantially by several orders of magnitude, both across temperature and for different variants. But the concentration of the molecules in the dense phase basically stays the same. That would rather be like saying that in a protein folding reaction, all of the interesting stuff was happening in the unfolded state, and they were all ending up in the same folded state, right? At least in the resolution of this measurement, that's what it looks like, right? I don't think that is what I'm advocating for, because it turns out the material properties are all quite different, and so is the spatial organization. But our ability to peer into these dense phases is still in its infancy.
Thanks very much. Rams, I'll turn it back to you. And thank you again for a wonderful talk and for your fortitude and stamina in answering the questions.
Thanks, Dan. Thank you very much, Rohit, for a great talk. I have a question, so maybe a very naive one. You have this competing intermolecular sticker interaction that deciding on the critical concentration above which a phase separation occurs. Let's summarize that way. In this case, what I wanted to say was, what happens if we have a metal which is bridging the stickers, forming metallocene-like scenario? Can it lead to phase separation, or can it lead to oligomer formation, or can it lead to, individualization of monomers as opposed
Absolutely. So in fact, the question you asked actually sort of in some ways gives me a hook for when I present this the next time. So essentially, the intramolecular interactions are setting the PU, right? So the concentration of unfolded molecules. The total concentration is setting the CSAT, which is, the sort of the overall concentration of these intermolecular sticker interactions are starting to now have a competing effect. The metals would now become a third player in that. So you would end up essentially now saying that, I'm generating, because they would bring de novo crosslinks, right? And so you would effectively say that I have created a new type of sticker. And that is exactly what I was suggesting in this paper we published last year. We show that if I have a ligand that is multivalent or bidentate or multifunctional, now I can start to add stickers or compete for stickers, right? So the whole idea is that in a stickers and spacers grammar, if the metal that you propose primarily coordinates spacers, then it's adding to the valence. But if it's engaging with stickers, but it's monovalent, then it's effectively taking away stickers. But if it's able to make bridges, then it's going to enhance phase separation. And you can actually work out just by how much, right?
So under what conditions or factors decide on oligomer versus phase separation,
Yeah,
or
so this is a really important, really important point. So phase separation is the result of non-stoichiometric interactions, right? Instead of making all of my complementary interactions between a pair or a small number of molecules, I, as a molecule, let's say I have 10 stickers, I would like to engage with 10 different molecules, right? That non-stoichiometric effect basically requires two things: multivalence, and that the interactions not be super strong. Right? Vis-a-vis KT, if there are 10 KT, then essentially what you're going to get are what are called network terminating interactions, right? And so super strong interactions will just basically give you very sort of well-defined stoichiometric complexes. The properties of the spacers also matter in that regard. They have to be of the requisite length, but also have a solvation preference that they themselves are not super sticky. So the poly Q, for example, really doesn't fall into the stickers and spacer because it is just poly sticker, right? Everywhere I look at, just have a sticker. Whereas I don't want to have spacers that are highly solvatable, because then what I will do is essentially the spacers are like sticking their elbows out and keeping stickers from coming together, right? So essentially the spacers need to be nature's instantiation of what I call Flory-random coils. Yep. They, in the competition between spacer-spacer, sticker-sticker, and spacer-solvent interactions, it doesn't care. It's like, all these interactions are good enough for me. I'm happy to mediate the stronger players, right? But if I now have spacers that are very rigid or very solvophobic or very solvophilic, now I'll start to sort of get these arrested phases.
Thank you very much.